Hi everyone, and welcome to our module on skeletal muscle. In this module we're going to talk about skeletal muscle, and cardiac and skeletal muscle are considered striated muscle, and the reason they're called striated is because you can see striations or little lines when you examine the tissue under the microscope. So for example, on the screen here on the left is a slide showing skeletal muscle, and I've zoomed in on the right so you can see the lines or striations. And this is in contrast to smooth muscle, which I talk about in another video. Smooth muscle is not striated. Skeletal muscle in your body is made up of bundles of muscle fibers, which are cells. They're narrow and long, and they contain structures called myofibrils, which are the contractile structures that allow muscles to work. They're called skeletal muscle because they attach to skeletal bones, and the attachment that's closest to the spine is always called the origin of the muscle, and the attachment furthest from the spine is called the insertion of the muscle. Let me define some of the important vocabulary that is used to talk about skeletal muscle. A skeletal muscle fiber is the same thing as a muscle cell. When someone refers to the sarcolemma, they are talking about the plasma membrane of a muscle cell. Hundreds of years ago, there were two fields of medicine. Osteology was the study of bones, and sarcology was the study of soft tissues. And that word sarco still persists, especially around muscle tissue, and the sarcolemma is just another name for the plasma membrane. Myofibrils are the contractile structures within the muscle cells, and the T-tubule is an invagination of the plasma membrane. Muscle cells have these little invaginations that look like this, and they look like a T, so they're called a T-tubule. The sarcoplasmic reticulum is an intracellular structure found inside of the muscle fibers or muscle cells. It's similar to the endoplasmic reticulum in other cells, and it's very important for calcium storage, as we'll see in the next few slides. The terminal cisternae are the sarcoplasmic reticulum portions that are right next to the T-tubule. So if this is a T-tubule here, some of the sarcoplasmic reticulum abuts the T-tubule, and that's called the terminal cisternae. And then finally, a triad is a structure found within skeletal muscle that consists of a T-tubule with cisternae on either side. Here's a picture from the web of a skeletal muscle fiber. So this entire structure is a muscle cell. This is the nucleus down here. Some mitochondria are highlighted here, and the membrane of the cell is called the sarcolemma. And you can see that it's a bundle made up of these myofibrils, which are the structures that contract in skeletal muscle. And here I've zoomed in on a myofibril. The pink structure in the middle here is a T-tubule. It's just an invagination of the sarcolemma or the plasma membrane. The structure shown in blue or purple is the sarcoplasmic reticulum, and you can see that the sarcoplasmic reticulum abuts the T-tubule on both sides. The portion of the sarcoplasmic reticulum that abuts the T-tubule is called the terminal cisterna, and the three structures together, the two terminal cisterna and the T-tubule, are called a triad. So now we've talked about how you have a muscle fiber, and within that fiber you have little bundles called myofibrils. Now what we're going to do is zoom in on one of those myofibrils and talk about the structures inside there. And we're going to start by talking about the sarcomeres. These are contractile structures within myofibrils, and they contain filaments, which are long stringy things called actin and myosin. The actin filaments tend to be thinner, so they're called the thin filaments, and they're called actin filaments because they are polymers of the protein actin. We'll see some pictures of those in a minute. Myosin forms thicker filaments, so the myosin filaments are called thick filaments, and they are composed of the protein myosin, and they have different domains called the head and the tail, which we'll talk about in a few slides. Besides the actin and myosin filaments, there are other structures in sarcomeres that you need to be familiar with. The Z discs are found at the end of sarcomeres, and these are like scaffolding that provide mechanical stability to the sarcomere. The Z discs contain filaments themselves called vimentin and desmin. These aren't really important to the function of muscle tissue, but you need to know that these are present in the Z discs because they're sometimes used as tumor markers to identify tumors that derive from muscle tissue. And I talk more about vimentin and desmin in the cell biology section in the video on the cytoskeleton. And then finally, titin is a cytoskeleton protein that tethers myosin to the Z discs, and we'll see a picture of this in the next slide. Here's a drawing of the sarcomere in the relaxed state. The blue structures here are the actin filaments, the red structures are the myosin filaments. The myosin tails are towards the center, the heads are these little red circles towards the sides. The Z discs are thick structures that serve as scaffolding on the sides, and then these are the titin proteins that connect the myosin filaments to the Z discs. And when you look at the sarcomere under the microscope, you can identify a number of bands and zones. So the I band is at the edge. It consists mostly of actin, but also the titin proteins connecting to the Z disc. The H zone is in the middle. This is the area where actin is not overlapping myosin. And then finally, between the two I bands, you can identify the A band. And when the muscle contracts, it takes on this form shown down at the bottom. So now we have more actin and myosin overlapping, What's happened is that the H zone in the middle has shrunk, the I bands have shrunk, but notice that the A band has not changed in size. The A band is the same size in the relaxed or contracted state. And then finally, one other structure that you should be aware of is the M line. That's this line right down the middle of the sarcomere. On this slide, I've summarized the key portions of the sarcomere. The I band is the light band near the Z discs. It contains mostly actin. The A band is between the I bands. It's where actin and myosin overlap, and there's no change in the A band with contraction. The H band is at the center of the sarcomere. It contains myosin only and no actin, and it shrinks in size with contraction. And then finally, the M line is where central proteins are found for alignment and stability of the sarcomere. And here's an electron micrograph of a sarcomere. You're often shown these and asked to identify the structures I just pointed out in the drawing 2 slides ago. So the easiest one to find is the Z line. Those are the thick lines at the edges of the sarcomere. Then it's also easy to find the M line, which is right in the middle. And once you've found the Z line and the M line, it's very easy to identify the A band and the I bands, as well as the H zone in the middle. So now we've talked about how a muscle cell, which is also called a muscle fiber, contains bundles of myofibrils. And we've talked about how those myofibrils contain sarcomeres. Now what we're going to do is drill down even further and talk about the individual elements of the sarcomere. And we're going to start by talking about the thin filaments and the thick filaments. So I told you that the thin filaments are made up mostly of actin, and that's true, but they also contain 2 other important molecules, troponin and tropomyosin. The thick filaments are made up of myosin, but remember that it's got a head and a tail, and it's the myosin head that binds to actin to cause contraction. Here's a drawing from the web to make this very clear in your mind. So this is one of the thin filaments, and it's mostly made up of these little balls, which are actin proteins. But it also contains this stringy thing, which is tropomyosin, and then these little tiny circles, which are troponin. Then the thick filaments, which are the myosin filaments, look like what's drawn at the bottom. It has various portions, but the most important is the myosin head, which is this little circle here. That's what's going to bind to actin and cause contraction. A brief word on the troponin element of the thin filaments. Troponin is actually a complex of 3 subunits. Troponin C binds calcium, the C is for calcium. Troponin T binds tropomyosin, the T is for tropomyosin. And troponin I is inhibitory. It inhibits myosin binding to actin. And even though we're talking about skeletal muscle in this video, you can also find troponins in cardiac muscle. And you may know that you can measure the blood level of cardiac troponins and use that to diagnose myocardial infarction. And I talk about this in some of the cardiology videos in more detail. Muscle contraction is initiated with calcium. We'll talk about where the calcium comes from a little bit later. Tropomyosin, which is part of the actin filaments, blocks the binding groove in actin for myosin so that myosin cannot bind and cause contraction. When calcium binds to troponin, however, the calcium-troponin complex removes the tropomyosin block. This results in a conformational change in tropomyosin, and this allows myosin to bind to actin. For this reason, skeletal muscle contraction is said to be thin filament regulated. This is because it's regulated by tropomyosin, which is part of the thin actin filaments. And this is in contrast to smooth muscle, which I talk about in another video, which is thick filament regulated. In the resting state, myosin binds to ATP and hydrolyzes it to ADP and a phosphate. When it does this, it assumes a cocked position, which means it's ready for contraction. When that tropomyosin block gets removed, myosin binds to actin. It then moves along the actin filament. This is called the power stroke. And after this, myosin binds a new ATP and gets ready to do the same thing again. Here's a picture from the web to make this very clear. This is the thick myosin filament at the bottom. This is the thin actin filament at the top. The myosin head, shown here, binds to ATP. It then hydrolyzes ATP to ADP and phosphate and assumes the cocked position shown at the bottom of the screen here. When calcium comes along and binds to troponin, it removes tropomyosin from the binding groove. The myosin head can then bind and form a cross bridge. It then goes through a maneuver called the power stroke and releases ADP and phosphate. And now what you have in the big picture is that myosin has bound to actin and contracted and moved down the actin filament. And here's a summary slide just to help you keep straight what's going on during contraction in the thin and thick filaments. So in the thin filaments, calcium is binding to troponin C. That then removes tropomyosin from the binding groove. On the thick filament side, myosin binds ATP. The myosin head becomes cocked as ADP is hydrolyzed to ADP, and then the myosin can undergo the power stroke once tropomyosin is removed from the binding groove. And here's a picture from the web just to summarize everything we've talked about so far regarding skeletal muscle. So if you look at the skeletal muscle, for example, the muscle in the shoulder of this man running, you find muscle that's attached to bone, and it's made up of lots of different muscle cells or muscle fibers. Within each muscle fiber, you find those little bundles called myofibrils, and one of them is shown here in this picture. And then if you zoom in on a myofibril, you find sarcomeres, and then if you zoom in on a sarcomere, you find actin and myosin. Now let's talk about the action potential of skeletal muscle. An action potential means a change in membrane voltage. I'll talk about what that is in a second. And this is required for skeletal muscle to contract. So let's imagine that we have a muscle cell and we have relatively more positively charged ions inside the cell and relatively more negatively charged ions outside of the cell. We would say that the voltage across the membrane of this cell is therefore positive because there's more positive charge inside the cell. Now let's imagine that we have another muscle cell and it's got relatively more negatively charged ions inside the cell and relatively more positively charged ions outside the cell. We would say that this muscle cell has a negative membrane voltage. And it turns out that muscle cells can go back and forth from a positive to a negative voltage, or they can go from a mildly positive voltage to a much more positive voltage. In other words, the voltage across the membrane can change. And there's a characteristic way it goes up and down, and that's called the action potential. So if you study the voltage across a skeletal muscle cell as it contracts, you would look over time and you would see that the voltage follows a pattern like what's shown on the screen here. It goes up and down, and that's the action potential of skeletal muscle. And this change in membrane voltage must occur in order for the muscle cell to contract. In other words, if you were monitoring time, you would see the membrane voltage change like this, and then a short time later you would see the muscle tone go up and down. The contraction is linked to the change in membrane voltage or the action potential. And this linkage between contraction and change in voltage is called EC coupling, or excitation-contraction coupling. In other words, contraction of the muscle via calcium is linked to the action potential, and contraction only occurs when the cell depolarizes and the membrane voltage changes. And the way this works is shown on the flow chart on the screen. So a neuron depolarizes that innervates a muscle cell. The neuron releases acetylcholine into the synapse. There are acetylcholine receptors on the muscle cells, and they cause the muscle to depolarize, and that leads to contraction. And here are some pictures from the web just to make this more clear. This is a muscle fiber or cell. This is the sarcolemma surrounding it, and this is a neuron that connects to the muscle cell via a neuromuscular junction shown here. Neurons, as you may know, have their own action potential, and when the neuron depolarizes from an action potential, calcium enters the neuron, and calcium triggers the release of acetylcholine, which is stored in vesicles as shown in the picture. The acetylcholine is released into the synaptic cleft, and then there are nicotinic acetylcholine receptors found on muscle cells at the other side of the synapse. This is called the motor end plate. When acetylcholine binds to its receptors, that opens sodium channels and allows sodium to rush into the muscle cell, and sodium is positively charged, so that will raise the membrane voltage, and that's how you get that upswing in the action potential. So this is what the skeletal muscle action potential looks like if you plotted voltage along the y-axis and time along the x-axis. The rise occurs because of sodium entering the muscle cell. The decline in the membrane voltage happens because potassium is pumped out of the muscle cell. And then, as I told you before, shortly after the membrane goes through this change in voltage, you will see contraction of the muscle cell. So the action potential is a change in voltage across the cell membrane. And remember that the cell membrane of a muscle cell is called the sarcolemma. So when the voltage goes up like this in the action potential, that's called depolarization of the sarcolemma. And when the sarcolemma depolarizes, the T-tubules will depolarize, because remember, the T-tubules are just part of the sarcolemma. When the T-tubules depolarize, as we saw earlier, they are right next to the sarcoplasmic reticulum. So that will trigger calcium release from the sarcoplasmic reticulum, and calcium, as we saw earlier, leads to contraction. So now what we have to talk about is how we get from the T-tubules depolarizing to calcium release from the SR. And in order to understand that, we've got to talk about two things: the dihydropyridine receptor and the ryanodine receptor. Dihydropyridine receptors are proteins that span the gap between the T-tubule and the sarcoplasmic reticulum. They're called dihydropyridine receptors because they have 5 subunits, and one subunit binds dihydropyridine drugs. Let's take a look at the bottom of the screen. The structure on the left is a pyridine ring. If you add 2 hydrogens and eliminate a double bond, you get a structure called a dihydropyridine. And you may know that there are drugs used in the treatment of hypertension called dihydropyridine calcium channel blockers. For example, amlodipine is one of these drugs. And it's called a dihydropyridine calcium channel blocker because it contains a dihydropyridine ring. Now here's the thing you have to understand, however, about skeletal muscle. These drugs have no effect on skeletal muscle. Even though they bind the dihydropyridine receptors, they bind with low affinity and do not affect the ability of skeletal muscle to contract. Their main effect is on smooth muscle, which I talk about in another video. Dihydropyridine receptors are also L-type calcium channel blockers, meaning they are capable of conducting calcium. But another confusing part about these proteins is that the ability to conduct calcium has nothing to do with their role in skeletal muscle contraction. Their only role in skeletal muscle contraction is to undergo a conformational change. Remember, they're proteins, and they change shape with depolarization. And when they do this, they open the ryanodine receptor found on the terminal cisternae, which is part of the sarcoplasmic reticulum, and this is their role in skeletal muscle contraction. Ryanodine receptors get their name because they bind ryanodine, which is a poison found in plants, but ryanodine has no role in the physiologic function of these receptors. This is just a confusing part of how these receptors got their name. Just like the dihydropyridine receptors, these are just proteins. They're large proteins. They're embedded in the sarcoplasmic reticulum, and the job of the ryanodine receptor is to release calcium and initiate contraction. And these ryanodine receptors are opened by the dihydropyridine receptors, so the two work together to release calcium and cause contraction. If we go back to this picture showing a triad in a muscle fiber, we have a T-tubule surrounded by two terminal cisterna of the sarcoplasmic reticulum. The dihydropyridine receptor spans the gap between the T-tubule and the terminal cisterna. It undergoes a conformational change when the T-tubule depolarizes, and when it does, it opens the ryanodine receptor on the terminal cisternae, and that dumps calcium into the muscle fiber. In addition to the ryanodine receptor, there's another structure in the membrane of the sarcoplasmic reticulum called the SERCA. The SERCA is the sarcoendoplasmic reticulum calcium ATPase. Its job is to transfer calcium from the cytosol back into the sarcoplasmic reticulum, and it's an ATPase because it uses ATP hydrolysis to function. The reason this is important is because if you want your muscles to contract quickly over and over again when you're exercising, you need to not only dump calcium from the sarcoplasmic reticulum, but you need to take it back up very quickly. And the reason you're able to do this is because of the SERCA. There's a very high yield clinical tie-in of all this muscle physiology, and that is malignant hyperthermia. This is a rare, dangerous reaction to anesthetics, especially halothane and succinylcholine. It's caused by muscle damage where muscles spill creatine kinase and potassium into the serum, so you can detect high levels of CK and hyperkalemia in patients with malignant hyperthermia. They present with fever and muscle rigidity after surgery where they were exposed to one of these agents. The cause of this condition, and this is what you need to know for step one, is abnormal ryanodine receptors. Some patients have abnormal ryanodine receptors, and if they are exposed to one of these drugs, they are vulnerable to malignant hyperthermia. The abnormal ryanodine receptors release excessive calcium, and then ATP is consumed as the sarcoplasmic reticulum reuptakes calcium, as we just discussed. The consumption of ATP generates heat, and that leads to muscle tissue damage, and that's what leads to the high CK and hyperkalemia. It also leads to the fever and the muscle rigidity. And you can treat it with a drug called dantrolene, which is an inhibitor of the ryanodine receptor. Dantrolene is a muscle relaxant. It's an antagonist to the ryanodine receptors. It blocks the release of calcium from the sarcoplasmic reticulum through the ryanodine receptors, and this reduces calcium in the cytoplasm for contraction. That's why it's a muscle relaxant. And its role in malignant hyperthermia is to stop those abnormal ryanodine receptors from dumping calcium into the muscle fiber. And this is a patient undergoing surgery to remind you of the type of patient who's likely to have malignant hyperthermia in the post-op setting. The last topic for this video is to talk about different types of muscle fibers or muscle cells. There are two types of muscle fibers, so-called slow and fast twitch. The slow twitch fibers get their name because the time to peak tension is long or slow. They're also called red fibers because they have a deep red color, and that deep red color comes from myoglobin. These fibers have lots of myoglobin to bind oxygen, and this allows them to resist fatigue. So you can imagine that these are useful fibers to have in muscle tissue beds that need to remain contracted for a long time. And a good example of that are the postural muscles of your spine. If you want to stand for a long period of time, like this man shown in the screen here, you need to stay contracted in terms of your spinal muscles and your leg muscles. So you will find more slow twitch fibers in your postural muscles. These fibers also have more mitochondria. They can perform more oxidative phosphorylation and more fatty acid metabolism, and they have moderate glycolysis activity. The other end of the spectrum are the fast twitch fibers. These fibers get their name because the time to peak tension is fast. They're also called white because they have a relatively pale color because they have less myoglobin. These fibers primarily metabolize glucose and glycogen. They have lots of glycogen storage, and they have increased activity of glycolysis enzymes. They have relatively few mitochondria, so they do less oxidative phosphorylation. The goal of these types of fibers is to do a quick on and off contraction. And for example, many of your eye muscles have lots of fast twitch fibers because their job is to just move quickly to one side or the other. And most muscles in your body are a mixture of fast and slow, and different people have different mixtures in their different muscle beds. And that concludes our video on skeletal muscle.